Good morning, good afternoon, or good evening, everyone. I'm Ron Vale, and I'd like to welcome everyone to Life Science Across the Globe. We're in our second round of talks, and today we're in Europe with our sister institute, the European Molecular Biology Laboratory, which actually has 6 sites across Europe. And we'll hear from individuals representing 3 of those sites today. So right now I'd like to introduce Cornelius Gross, who is the interim head of EMBL in Rome. And Cornelius is a neurobiologist who studies neural circuits involved in fear and anxiety. And he also studies how microglia contribute to brain development. So Cornelius, welcome.
Hi, thanks, Ron. So it's my pleasure to introduce 2 speakers from EMBL for today's talks. In case you're not aware, EMBL stands for European Molecular Biology Laboratory. And as you may know, EMBL is an intergovernmental organization. It's funded by member states across Europe. And it has a mission to carry out basic research in molecular biology and to train and to support scientists across the globe. And as Ron mentioned, we operate at 6 sites across 5 countries in Europe. So for today's first talk, for the science talk, it's my great honor to introduce Janet Thornton. Professor Dame Janet Thornton is a renowned scientific leader whose early career was rooted in structural biology and who is widely credited alongside with others for the establishment of a new scientific field of bioinformatics. She studied physics originally, and then she completed her PhD in biophysics at the National Institute for Medical Research in the UK. And then after postdoctoral research at Oxford University, she took up professorships at Birkbeck College and University College London. From 2001 until 2015, for 15 years, she was the director of EMBL's Research Center in the UK, the European Bioinformatics Institute, and she continues her research there now as a senior scientist. She is both a commander of the British Empire and a Dame of the British Empire, and these awards were conveyed to her for her services in bioinformatics. She's a fellow of the Royal Society and vice president of the European Research Council. Her research has focused on the impact of genetic variation on protein structure and disease. And her talk today is entitled Computational Enzymology, the Structure, Function, and Evolution of Enzymes.
Thank you very much indeed, Cornelius. And it's a great pleasure for me to be here this morning and to speak to everyone all around the globe. It's very strange, and I hope this will work okay. So I'm going to share my screen and tell you about computational enzymology, which has become very important in my research. And in particular, I'll talk about the structure, function, and evolution of enzymes. Oh, hang on. So, what are the outstanding questions in enzymology? Enzymes have been studied for many, many years. But there's still many things we don't understand fully. We don't understand how many enzymes work. We don't understand how some functions, new functions evolve. We really cannot yet predict function from sequence and structure, and neither can we predict transformations and mechanisms. And these, to me, are all very strong academic goals to work towards. But of course, understanding enzymology and being able to predict has huge applications in biotechnology, medicine, and agriculture, such as in designing new enzymes like plastic processing ones, or redesigning pathways to make new molecules. So our goal is to capture, to analyze, to validate, and ultimately to predict enzyme mechanisms, and to discover how enzyme function in a family changes during evolution. So the talk will be in 3 parts. the evolution, the enzyme data, and transformations and mechanisms. But first, the reason that we got involved in functions was because of a classification of enzyme functions that's been done by the Enzyme Commission over over 50 years now, manually. And each enzyme is given a number that defines the reaction that you can see, I think, here at the bottom. It says nothing about the protein that does this reaction. So it just defines, if you like, the small molecule reaction or large molecule, depending on what it is. So this classification has 4 numbers. The first one, the C, is the primary classification. You can see here the 7 different classes of enzymes. There are many databases that define other parts of reactions. Probably the best known is the KEGG database from Japan, but also many others that work to provide additional information. But we were interested some years ago now in understanding how enzymes evolve new functions. So in a family, new members of a family are generated, and very often they may adopt different functions, or indeed they may become pseudoenzymes and not functional catalytically at all. So what we wanted to do was a computational analysis, really to provide an overview of our current knowledge of the observed functional changes in enzymes. So we used public data, sequences, structures, and functions. We've honed in on those sequences which were known to have different functions, and we measured that by changes in the EC numbers. And we basically chose to identify those associated with novel functions, and we focused on the small molecule metabolism. We only used experimentally validated functions, and we omitted pseudoenzymes or non-enzymes because of the complication with the data. So let me give you an example. Here we've got one family that I'll call PIPDE, the superfamily. It's a very large family. There are many thousands of sequences in the databases. This family occurs in many species, and we ourselves have over 26 examples in our genome. There's lots of structural data, and we also know that this family has enzymes that performs at least 18 different enzyme functions. This is one of these particular enzymes, PLC, phospholipase C, and this enzyme cleaves the OP bond in this molecule here with the help of a magnesium ion as a cofactor. But if we look at the whole evolutionary tree for this family, what we see is captured here by the EC numbers is different EC numbers, i.e. different functions. And there are 3 that are highlighted in pink and in blue. And those you can see here superposed. Their structures are superposed. You can see very easily they're evolutionarily related. They belong to the same family. And you can also see that they perform, in this case, the hydrolases, and in this case, the lyase. And you can also see that they operate, from the name of the enzyme, they operate on very different substrates. And so from this tree, we can build a functional changes matrix. And this, we call it the EC exchange matrix. And the idea is that we count the number of times there is a change in function. So here we've got, and you can see here on the phylogenetic tree, there's been a change between 2 lyases. Here you can see there's been a change between a lyase and an isomerase, and a hydrolase. And there are 4 examples where we have hydrolases changing into other hydrolases with different substrates. And so we can construct for every family where we have a phylogenetic tree and different functions, we can construct this EC exchange matrix that captures how often enzymes change their function. And so we can, by looking at, we looked at over 276 superfamilies and put together, we added up all these functional changes to look to create this overall EC exchange matrix. And this matrix, which you can see here more clearly, highlights in numbers, you can see 1212 and 42 and 28, the number of changes in enzyme function that were known at the time we did this study. And what you can see is that every box is occupied. There are changes from all classes to all other classes, but the majority of changes occur along the diagonal here. And so what we found was that the number of changes in substrates, i.e. within the same primary EC class, constituted about 89% of all the changes that we saw. And in contrast, the changes between enzyme commission classes constituted only about 11%. And so from this, it's very clear that during evolution, an enzyme changes its substrates or within an enzyme family much more frequently than the changes in the chemistry, the changes in the reaction chemistry performed. But we also looked at how often you got changes from one class of enzyme to another. And Sergio looked at the isomerases in great detail. And what he represented here by these blue arcs is the changes that we observed, and the size of the ribbon constitutes the number of changes, so that you can see you get just a few changes between isomerases and ligases, but rather more between isomerases and transferases. And in fact, when you look at all the 6 classes of enzymes as there were at that time, you can see all changes are possible, basically. And so we can see that changing enzyme function occurs very often during evolution, not often in terms of timescales, obviously, but there have been many, many examples. And that, in fact, when we look at the enzymes that are today, most of those have evolved from enzymes that originally performed different functions. And so we thought we would think about this theoretically. And you will probably be, I hope you will be aware that you can map how a protein structure changes as the sequences diverge. So you can see here the sequence identity. So here we're comparing 2 protein sequences. And as the sequence identity drops between any 2 proteins, the structural similarity between those proteins falls. And you can see the curve here that was originally derived by Chothier and Lesk, showing as sequence diverges, structure diverges. So in our mind, we were interested to ask what happens as the sequence between 2 enzymes drop? What's the functional similarity between that? And this was the question we were asking. And we sought first to look at specificity. Now, this is just one aspect of enzyme function, but it's probably the one that's most easily captured computationally. So our aim was to compare substrates of domains as their sequences evolve. So we could use standard sequence comparison tools to evaluate the sequence similarity between 2 enzymes in the same superfamily. And in terms of the substrates, we can use a Tanimoto or Jacquard coefficient to measure the similarity between the small molecules. And the sorts of changes on the graphs that, well, let me go to the next slide. So essentially within a superfamily, we create subfamilies with the same function, and then we compare these 2 subfamilies and we calculate an overall average sequence similarity, and standard deviation between the sequence. And we also calculate the substrate similarity, the reactant similarity. And so we can draw a plot saying, how does substrate similarity and sequence similarity change for a given superfamily? And what we expect is that as the sequence similarity drops, then we expect this protein will have diverged further and the substrate similarity will similarly fall. So this is what we were wanting to explore. So the results perhaps didn't turn out quite as we hoped they might do. This just shows one example to give you a feel. So here we've got one particular domain, and we use the Cath domain from Christina Rengo's group at UCL. And we map for a given family, like here, each point compares two subfamilies with different functions within this large superfamily. And what we can see is that as the sequence drops, as these sequences diverge, then here for this two families, we can see that their substrate similarity is very different. Whereas if we move up to here, where it's a higher sequence similarity, then the substrate similarity is similar. And although this clearly is not, I would say, a linear plot, we drew a line to give us a feel for the changes that we were observing. But you can see the correlation at R² of 0.19 is very low. You can also see, and this is the colors, the red families correspond to changes in the primary class. The blue and the green and orange correspond to changes of subclasses or sub-subclasses. So these red changes are more extreme than the blue, green or orange changes. So for this subfamily, as the sequences diverge, the substrates become more different, and so do the functions. If we now look at another family, and you can see in this family, the large alpha beta family, Cath code 3202060, we can see in this family, this shows all the changes in EC numbers that we observed within this superfamily. And then we plotted again, as we did before, and now you can see the curves are all over the place. The plots are all over the place. And so it appears for this superfamily that there's absolutely no simple relationship between divergence of sequence and structure and functional divergence. And in fact, we had the data for our 276 superfamilies, so we were able to plot these lines. And what you can see here is one example. We were hoping to have some nice correlation, and you can see on average, there is indeed, as the sequences diverge, the substrates diverge. But you can also see that there are families that are all over the place with many different scenarios. And so it's very clear that there is no simple correlation between sequence identity and the functional similarity as measured by the similarity of the substrates for all families. So this made us perhaps think again. And so really we have gone back to basics in trying to look in more detail at enzyme catalytic sites and enzyme mechanisms. And this we started many years ago, and this has evolved into a data resource that's called MCSA. And this data resource has information not like the EC nomenclature that just describes the reaction. This obviously has the reaction. It also has the enzyme sequence. It has the cofactors that are involved. And it also has the catalytic residues that form the catalytic site. But as well as that, because in order to know what the catalytic residues are, you need a reasonable knowledge of the mechanism. And so we also extracted from the literature, we take the mechanisms proposed from the literature. And these are captured, they're not just captured as curly arrow diagrams like you can see here, but they're captured as smart patterns that we can actually begin to compute on. And so we decided we would look in more detail because, of course, now we have quite a lot of data about enzymes. In total, we have about 684 mechanisms. We have 3,300 odd residues, catalytic residues. We have the mechanism steps in the enzyme as it proceeds through its reactions. And so we wanted to really capture this knowledge so that we could link the residues and the chemistry of the protein to the chemistry of the reaction. And the hope, of course, is that this knowledge, as we gather it, will help us predict mechanisms and do de novo enzyme design. And so the very simplest thing that one can do is ask which are the most common catalytic amino acid side chains. And what you can see here is that histidine, asp, glue, lyse, arg, the charged amino acids dominate enzyme catalysis, as you would expect, and they are followed then by the polar and aromatic amino acids thereafter. So these are reactions that are happening through the side chains of the amino acids. So the charged are more common than the polar side chains, are more common than the hydrophobic side chains. This has been known for some time. We can also look at the main chain because, of course, every residue has a carbonyl and amino group in the main chain, and they are sometimes, not very often, but sometimes involved in catalysis directly. And what you can see here is that in this case, in fact, glycine is the most common residue, followed by serine, alanine, cysteine and threonine. And clearly it's the small amino acids that allow, if you like, the substrate access to the main chain that explains this data. But we wanted to go farther. We wanted to look at what the actual amino acid roles do in the enzyme reaction. And so we defined residues as being either reactants, spectators, or interaction residues in catalysis. So we used this quite complicated ontology called EMO to assign a role to each amino acid. And what we found, we could then say, well, which are the most common reactant roles? And what we found was that proton, acceptor, and donor, that you can see here, completely dominate enzyme chemistry, completely and utterly. In terms of the interactions, we find hydrogen bond donor acceptors and metal ligands are the most common. And in terms of spectators, as you would expect, electrostatic stabilization is by far the most common role for a spectator residue. But we can go even farther because we can then say, well, of the 20 amino acids, which role do they each play? So this shows a sort of a heat map or a distribution of roles played by the residues. And these residues are in order of their use as catalytic residues in our enzyme data set. And you can see, first of all, that of course the charged and polar residues at the top here are the proton acceptors. You can see that the electron pairs at the bottom here, the single electron acceptors and donors is performed by more or less all the amino acids. And you can also see that cysteine, which is a very common catalytic residue, is actually a very versatile residue because it can perform all of these different roles, especially associated with the radicals. And so we can look at nucleophiles in more detail. And if we look at the nucleophilic residues, the ones that act as nucleophiles in the enzyme reaction, we find that there is specificity for different amino acids for the different substrates or reaction centers that they act on. So serine, for example, we find here can act on several residues, but we find cysteine and serine act on one, on the carboxamide, whereas lysine acts on the iminium in the PLP cofactor. And so what we can find is that serine, the most common serine electrophile, are the amides, of which there's 21. And of course, this is the mechanism used in serine proteinases. But we can also capture the other roles. And so with this provides us with, if you like, detailed information about the role of different amino acids in enzymology, in catalysis. And so we've captured this in a way that we can compute on it. And so we can begin to look at, and we can begin to compute on computational transformations by enzymes. And I don't have time today to go into this in detail. It's work by John Tyzak, a very talented postdoc. And he's developed something called Transform Minor, which allows us to actually map enzymes which perform catalysis on similar substrates. So you can find enzymes that will act on a source molecule that you specify, or alternatively, if you want to convert one molecule to another molecule, you can find the pathway from that molecule to its target. And there may be a direct route, but there will also be other pathways. So for transformation, this is the tool that we are developing. And the hope is to use this actually for enzyme modification and repurposing. And we're in the process of trying to experimentally validate this in collaboration with partners. But the other real basic thing about enzymes is their mechanism. And we want to try to predict enzyme mechanisms if we can. So if we know of reaction, and you can see here, this is an isomerase from glutamate, that reaction proceeds through 4 steps that are shown here, where the sort of classic curly arrow diagram that drives the conformation. And so the goal that we have, and we are not there yet, I'm just telling you about our current work, is to create an algorithm that, given an active site and the reaction of an enzyme, can we suggest or predict possible enzyme mechanisms based on the information that's stored in MCSA. So this is an informatics and knowledge-based approach to enzyme reactions. And so our goal is to extract rules from the MCSA, to then create an algorithm that can apply those rules and say where, develop the steps, present a results page, and then validate it on the data that we've got. And this clearly is an iterative process. We're at the stage where we have lots and lots of rules derived from all of the steps. And in fact, in total, if I just move this, we have combined rules, about 2,500 rules that can be used to map the transformations. And so we have parsed the data, we have our algorithm, and we're just working on these last 3 steps here. And it's clearly not, it's not possible to do this without quantum mechanics and molecular mechanics and quantum mechanics to really predict a mechanism. But it is possible, I think, for us to be able to suggest different mechanisms that can then be looked at with much more sophisticated approaches using quantum mechanical methods. And hopefully this will enable us to improve the understanding of enzyme mechanisms. So that's really all I want to say. What we want to do is to understand and predict enzyme evolution. I'm not returned to this, but this is part of what we're doing and will involve the tools that I've described. We want to identify pseudoenzymes and create catalytic modules in 3D. A key challenge is to identify substrates and products for novel sequences. And this really, I think, at the moment is probably the key challenge. We want to predict transformations, we want to predict mechanisms, and ultimately use this knowledge to repurpose and redesign or design new enzymes. So that's really our vision for the future. I'd really like to acknowledge the people who've been involved in this. Gemma, Assad, Nick and Sergio were in the lab as postdocs for several years and worked on different aspects of this. At the moment, Antonio is working on the prediction of mechanism and John is working on the transformations. And this work is a collaboration with Christina Rengo's group at UCL and obviously all the rest of my group at EMBL. And I'd really like to acknowledge funding from EMBL and also from the Wellcome Trust. And I hope that I convince you that enzymology is actually a very exciting field and there are many things that we can still learn. And I welcome questions. Thank you.
Thank you, Janet. That was a really interesting talk. I think you did convince most of us of how exciting enzymology is and can be. We do have quite a few questions from the audience, but I want to start with sort of a broader question of, is your lab at all applying any of your computational tools to studying or modifying specific proteins or looking at specific diseases, or is your group more doing tool development for other labs to focus on these things?
So we really do both. And what I find is that different postdocs and different pre-docs have different inclinations. So some of them love the sort of computational programming aspects and like to develop new methods. And over the years, we've developed many methods that are freely available and are used enormously around the world. But in addition, there are people in the lab who are working on specific challenges, especially looking at variants and the structural impact of variants. And so we're looking at a variety of, in fact, developmental diseases and variants that are reported in the clinic and trying to explain those from a structural perspective. So we don't focus just on one. At the moment, in the enzyme world, we're beginning to look at triterpene synthases, which synthesize the triterpenes, which are incredibly important molecules for a whole variety of roles in biology. But that work is just beginning now. So I have to say, maybe it's my physics training. I tend to take a rather broad overview, but I'm equally aware that individual problems obviously are extremely important, and they're very good to test whether our methods are working or not.
Great. So we have a lot of questions about the similarity graphs that you showed.
The lack of similarity graphs.
Yeah. So the first is from Ratika Sood. Ratika wants to know, with the sequence and structural similarity curves, can you comment on why it wouldn't be a linear relation between sequence and structural similarity?
Well, the sequence and structural similarity, we know, and this has been known for some time, that actually the sequence similarity can drop to about 40% similarity, identity, shall we say, rather than it's easier to understand, and yet retain the structure, almost all of the structure. So down to about 40% sequence identity, the structure is very conserved. There are insertions, there are new loops, the loops perhaps change their position, but the primary structure is the same. Once you drop below 40%, going down to 20% and even lower, many sequences are much lower, you see the conservation of the core of the protein structure. And for the enzymes, It's the catalytic core, which is extremely well conserved. Of all conservation in biology, the catalytic residues are more conserved than anything else. And so these are very special residues that really perform the catalysis. But of course, without catalysis, we wouldn't have any life. So it's really important to look at that in detail. But those changes, so what we see is those changes between class, primary class, enzyme class, really occur only at rather low sequence identity, often below 30%.
Right. So, you know, along those lines, have you or anyone else looked to see if functional similarity correlates with structural similarity?
So there is correlate and correlate.
Right.
It certainly isn't a linear relationship, and we all know, just as the change of a single residue can destabilize a protein, if you're looking in an enzyme binding site and you change one of the residues in that binding site, that can change the substrate specificity, even if the whole of the rest of the protein stays the same. But in general, evolution works, you know, most of evolution, I always think of creeping and leaping evolution. Most evolution creeps very slowly. And so that when you see a change in function, generally that happens at lower, you know, sort of 60, 50% sequence identity. Not always, there's huge variation. But because, and then if you have a major change, if you have a change in the chemistry of the reaction, that really is at these low sequence identities when you have a long time of divergence. And in fact, many of the changes in function that we observed in our big analysis clearly occurred in bacteria, many, you know, right at the beginning of the evolution of life. So trying to map all of these is quite complicated. It's somewhat hidden in the midst of time, you know, in the depth of time. But nevertheless, these changes were clearly critical to evolve life to be able to cope with the changing environment.
Also, another question that came in is it possible that the same structural domain leads to different functions in the same class of enzymes?
Oh yeah, well, so there's function and function. One aspect is there is function in a test tube.
Right.
There is function in vivo, and that will be different again. So the same, just we know serine proteinases are involved in all sorts of different biological functions, like you know, in the gut or in the blood, in the brain. So you have the biological function is different from the function in the test tube, if you like. But the key thing is that if you take, and we can see this, you take an enzyme and it is capable over millions of years of evolving new functions that do different things. Most often the functions are related, but not always. Occasionally completely new functions evolve. In contrast, you can take the same EC number, i.e. the same reaction, and you can find 2 or 3 enzymes that perform that reaction using completely different mechanisms. So it's almost evolution, you know, over 4.5 billion years, it can do wonderful things. And so you see almost every type of change that you might envisage.
Well, thank you, Janet. I think at this point, we are going to move on to our second speaker. And so I would like to turn it over to Lucia.